Hello everyone. Today we are going to discuss about the methods of bacterial growth measurement. Growth is defined as the increase in cellular constituents, and this can result in an increase in cell number or cell mass. Growth can be monitored by measuring cell mass or cell number. In bacteria, growth represents different phases. So there are four different phases for bacterial curve. That is the lag phase, exponential phase, stationary phase and death phase. Lag phase is called so because the growth is so much delayed in this phase and the cells are getting adapted to the new environment. That's why it is called as a lag phase. Once the cells are adapted, they start to grow exponentially. So this phase is called as the exponential phase or the log phase. Once the nutrients are utilized, there is no more add-on nutrients in the media. All the nutrients are used up by the bacteria and it reaches the stationary phase. Death phase is followed by the stationary phase. So once the nutrients are depleted and the toxic products are accumulated in the system, bacterial growth curve enters the death phase or the decline phase. So it is important to measure the bacterial growth and it is useful in various applications. Depending on the application, the different methods can be chosen. There are two main methods, direct method and indirect method. Direct method include plate count, filtration, most probable number and microscopic count, and in indirect method it is turbidity, metabolic activity and dry weight. So measuring all these parameters will help to identify the growth of bacteria. One of the accurate measurement for growth is the plate count. So in this case, the bacterial culture is diluted and plated on agar containing media. So the plating can be done by streaking or by port plate method. Once the culture is plated, that need to be incubated for overnight or 24 hours for the growth to occur, and the number of colonies that are formed in the plate is counted. Based upon the dilution, the number of colony forming units in the original sample can be calculated. Here, in this case, only the viable bacterial cells are counted. One of the disadvantages is that it takes almost 24 hours or more for the colonies to grow. Depending on the bacteria growth we are monitoring, it may take longer time. In plate count, both streak plate as well as pore plate can be used. In pore plate, the bacterial colonies are mixed with the agar containing medium under 60 degrees Celsius and that it is poured on the Petri plate. So here the problem is that the bacteria which is sensitive to temperature will not grow. It is not useful for the culture that are sensitive to temperature. The other method of bacterial growth measurement is filtration. So in this method, the bacterial culture or the media containing the bacteria is filtered and the filter paper is placed on a media in the Petri plate. Based on the growth, we can identify the number of bacterial cells. This is mostly used in the water samples where the concentration is required. The other method is the most probable number, which is shortly called as MPN technique. So this technique is used mainly to measure the bacterial culture that will not grow on solid media. So the sample is diluted repeatedly and inoculated on several growth tubes for each dilution, and the number of colonies are counted from each positive tube. The statistical method determines the 95% probability that bacterial population will fall in certain range. The other direct method is the microscopic count. In this case, the hemocytometer kind of slide can be used and the fixed volume of culture is added to that so that the slide will having different grades and the cells can be counted under the microscope. The advantage of this technique is there is no incubation required, but the disadvantage is that if you are looking for a growth of motile bacteria, it is difficult to monitor in this case. The other indirect method include turbidity, metabolic activity and dry weight. Turbidity is most commonly used in the bacterial culture as the bacteria grows and the turbidity of the culture media increases. Using spectrophotometer, optical density of the culture can be identified. A well-grown culture may require dilution. The advantage of this method is there is no incubation required. In this case, directly the culture media can be diluted and checked on the spectrophotometer. Generally, OD 600 is taken. The other method include metabolic activity. As the bacteria grow in the media, they produce certain byproducts or other metabolic compounds. So the measurement of such metabolic products can be used for monitoring the growth. The other method is dry weight measurement. The cells are centrifuged and the cell pellet is weighed. In this case, it will not distinguish the live and dead cells. Thank you.